Good afternoon, everybody. I've got 5 minutes for my presentation, so I'll rattle through it. My name's David Hughes. I'm a researcher at the University of Glasgow. The University of Glasgow is a beautiful campus. Unfortunately, we don't always have blue skies, although people would like to portray that. Typically, the weather is inclement, and the climatic environment, as well as the academic environment, is ideal for the type of work that I do, because I have a profound interest in understanding the circuitry of the dorsal horn of the spinal cord. And what we have in this panel over here are these blue blobs, and what these blue blobs represent are individual neurons. And these neurons come in a variety of shapes and sizes. They have various chemical signatures, and they also have various physiological signatures. And what we need to remember is that these individual entities are actually living entities, and they form dynamic processes for the transfer of information within discrete circuits. So what I want to try and do is to understand the individual components of distinct sensory pathways, to understand how they function in normal situations, and then to see how the properties of these cells change in conditions that develop chronic pain states. Now, chronic pain is a debilitating condition, as we've heard so eloquently this morning, and it forms a profound problem clinically, and because we essentially need to devise much better therapeutic properties to try and alleviate these debilitating conditions, because seemingly only 2/3 of chronic pain patients are currently responsive to the treatments that we have. So we still have a large population within our society that desperately need support. We can address this from various perspectives. We can use a scattergun perspective to try and work out globally, if we chuck on a particular drug and it affords some degree of pain relief, then that's great. But that kind of approach can be limited. The perspective that I'm taking for addressing neuropathic pain as a result of peripheral nerve injury is to try and identify individual components within functionally distinct circuits, and then to see how the properties of these individual populations change in allodynic animals. So quite a bit is known to some extent about the circuitry and the neuronal populations in the lumbar region of the spinal cord, spinal segments L2, L3, L4, because these are the segments that innervate the hind limbs, and these afford us fantastic targets to try and manipulate and induce different types of sensory responses. And because of our understanding of neuropathic pain using peripheral nerve injuries of these descriptions, I'd like to try and use this model to see if it actually works as a template for understanding visceral pain, because it's much more difficult to manipulate afferent fibers that innervate the viscera, and it's much more difficult to try and understand the central connectivity of the variety of peripheral of afferent fibers that supply the viscera to see what types of circuits that they actually integrate with. So as I've mentioned, I do work primarily on animal models. This affords us the ability to manipulate the peripheral nervous system and to then assess the behavioral consequences of those manipulations by testing their responses to applications of various types of stimuli. One of the most easily accessible models that we use is what's known as the spared nerve injury model, where we ligate and transect the tibial and common peroneal nerves. These are nerves that innervate a large portion of the hind limb, and we also keep what's known as the sural nerve intact. So essentially what that allows us to do is to find an area of mechanical hypersensitivity on the lateral aspect of the foot. And by using various behavioral testing paradigms, we can actually assess the impact of damaging these peripheral nerves selectively. Now that we have a behavioral readout of responsiveness to these mechanical stimuli, we can then try and understand what's actually happening to the pathways originating from the skin and projecting all the way up to the sensory cortex to ultimately allow the individual animal to perceive these different stimuli in a variety of different forms. So essentially what I need to do is to identify discrete neuronal populations from a heterogeneous mix of different types of cells within the spinal cord. And essentially what I try and do, that hasn't worked, is to strip away from this almost forest of complexity to identify individual neuronal populations and to see how they connect together. And we can define neurons on a variety of bases, including their morphology, the neurochemistry of the individual nerve cells, and also on their electrophysiological properties. Now, the spinal cord of the dorsal horn has a degree of organization to it, even though the component parts are extremely complicated, where the more superficial layers are responsive to pain afferent fibers projecting in and activating essentially pain circuits, whereas deeper layers of the spinal dorsal horn are involved in processing information from innocuous tactile stimulation. In chronic pain conditions, what happens in, for example, tactile allodynia is that these innocuous fibers in some way activate pain circuits to result in the perception of pain. To understand these pathways, we have to have an appreciation of the different types of primary afferent fibers that project into distinct regions of the dorsal horn. And historically, these have been accomplished by carrying out really, really challenging electrophysiological experiments where they impale individual sensory axons with sharp microelectrodes, be able to actually record the activity of these individual neurons by manipulating peripheral receptive fields, filling the cells with a neuronal tracer, and then recovering the morphology of these individual neurons. Now, these are heroic efforts, and people have been able to classify a whole series of different types of cutaneous sensory fibers based on their morphological properties. This gives us a basic overview, where essentially what we see is that pain fibers project in from the periphery and terminate within the superficial laminae of the spinal cord, activating pain centers, which is then relayed supraspinally. Whereas the innocuous touch fibers project in from the periphery and arborize within the deeper dorsal horn laminae to activate touch-specific circuits there. More recently, molecular genetic approaches have been used that allow us to identify these populations much more easily than carrying out these individual intra-axonal labeling approaches. And this has been revolutionary because it's allowed us to specifically pick out functionally distinct populations of afferent fibers, and we can then selectively manipulate their activity and also look at their central connectivity in a much more amenable way. One distinct advantage of this is that we can then try and adopt what's known as the Cree-Lox system, where we essentially have two types of reporter, or two types of mice. One where there's a sequence for Cree recombinase, which is downstream of a selective promoter. And this parent is then crossed with a reporter mouse line, where there's a reporter sequence for either a fluorescent molecule or a light-sensitive ion channel that's effectively silenced in this parent, but when the two are crossed, the Cree recombinase takes away the stop cassettes, and effectively any cell that has the promoter will express the reporter molecule. And that allows us to target individual neurons with great precision. In the spinal cord, there's a whole series of neurochemically defined populations of interneurons. Some markers are more ubiquitously expressed than others. So to get greater precision for targeting these neuronal populations, what we've recently been able to develop is intraspinal injections of various Cree-dependent viruses, which allows us to have greater selectivity of the timing for the expression of particular markers, because these markers can be expressed at different developmental time points, which will then ultimately which will basically reduce the fidelity of the expression that we're actually seeing within these mouse lines. So by adopting these approaches where we're using the insertion of light-sensitive ion channels, we can actually assess the behavioral consequence of selectively recruiting these types of cells. And these experiments from a group in McGill are able to show where we have photostimulation, which is light activation of skin receptors in a particular class of afferent fibers, we're able to actually show that recruitment of these sensory fibers will result in a pain-like withdrawal response. This doesn't cause any damage to the skin of the animal, but it does activate the first component of that pain pathway. Now, when we look in the central portions of the spinal cord, we can identify the great heterogeneity in the morphology of individual nerve cells. And this can be exemplified here using basic histological staining patterns where we have various morphologically distinct cell types in different dorsal horn layers. So that hints at a degree of functional specification for these morphologically distinct populations. By adopting an intersectional strategy to try and untangle this molecular heterogeneity that we have, we can target these neuronal populations much more specifically. For example, we can have Cre recombinase in one parent mouse, crossed with another mouse where there's a different recombinase, and then in the offspring of these animals, we can selectively introduce a Cre flip-dependent virus, which allows us to have an intersectional approach to target these neurons with much greater precision. And in doing so, we can actually understand much more accurately what distinct neuronal populations are actually doing. In this type of experiment, what we've done is to carry out targeted electrophysiological recordings from a GFP-expressing mouse line. And by targeting these cells in sagittal sections of the spinal cord, that is, slices of the spinal cord cut along the long axis, we're able to record physiological properties of these neurons and relate them to the morphology of these individual cell types. And what we typically see is that all the cells that have been highlighted in yellow here and outlined over here form a population of neurons that release glutamate. So these are excitatory interneurons. So we have distinctive electrophysiological firing patterns and distinctive morphologies for these excitatory interneuron populations. Whereas for the inhibitory populations, these have also a defining physiological and anatomical signature, and that they have high frequency action potential generation in response to currents, and what's known as islet cell morphologies, where they have an extensive dendritic arbor running across up to 400 microns along the length of the spinal cord. Now, these properties, addressing these populations can be very laborious, but recent technology has, and transcriptomic studies, have been able to identify distinct clusters of different neuronal populations based on the expression of various transcripts. And this work by Haring et al. Recently serves to form the opinion that these distinct subpopulations that can be identified using transcriptomics very often correspond to neurochemically distinct populations of neurons that have been identified using immunohistochemical approaches. So that gives us great confidence in that we're able to identify both excitatory and inhibitory subpopulations with great precision, which allows us to then try and understand the roles of these different neurons in normal sensory processing. In the deeper dorsal horn regions, we've also been able to identify very similar morphological and physiological properties for neurons that are involved in innocuous tactile sensation. And together, a recent review by Becky Seal has been able to try and amalgamate all this information so we've got a better understanding of the basic circuits and the way that they actually connect together. But the main question is, what changes in chronic pain states? What components of these circuits change? There have been various hypotheses. Shame this should be animated, but unfortunately not. One hypothesis has suggested that these innocuous touch myelinated afferent fibers that terminate in the deeper layers of the dorsal horn, after peripheral nerve injury, they extend or sprout dorsally to engage with and activate pain circuits. Unfortunately, we found evidence to the contrary, so that's still out there for discussion. Another proposal was that these myelinated afferents after peripheral nerve injury now tend to upregulate substance P and CGRP. They don't normally contain these peptides. These peptides are normally expressed in pain fibers that terminate in the more superficial laminae. But this hypothesis suggested that after damage, the tactile afferents now express this peptide, And when you activate them, they release the peptide, it then diffuses across the laminae to activate pain circuits. Once again, unfortunately, we found evidence to the contrary. And the final hypothesis for tactile allodynia that I'd like to present is that after peripheral nerve injury, it was proposed that there's a selective loss of inhibitory interneurons in somatotopically distinct regions, hence the skull and crossbones. Once again, we found no evidence for that. So we have to come up with new ideas. We've challenged various proposals, and I think it's fair to say that we, about time, have to come up with our own ideas. One thing that we've reported very recently is that a neurochemically distinct populations of neurons that sit in lamina 2 inner and 3 that express the calcium-binding protein parvalbumin. These form what's known as axonic synapses, so they gate the passage of low-threshold information into the spinal cord directly, but they also inhibit a population of excitatory neurons called vertical cells. And these vertical cells essentially have the anatomy that would allow them to act as a conduit for low-threshold information into the deeper dorsal horn layers, transmit it into lamina 2, and their axons then project up and engage with pain circuits. Now, under normal conditions, these parvalbumin-expressing inhibitory interneurons gate afferent input from low-threshold mechanoreceptors and also inhibit the activity of vertical cells. But after peripheral nerve injury, we found that the inhibition that these cells mediate is drastically reduced, which then opens this gate, allowing information from the low-threshold tactile fibers to be projected through the vertical cells and to activate the lamina 1 projection neurons. So that's a role for inhibitory interneurons in chronic pain states. And as Steve mentioned earlier on, a degree of central sensitization or increased excitability of dorsal horn interneurons is an important feature of the development of chronic pain, which is why we've looked at this population of excitatory interneurons, the calretinin expressing interneurons. And again, these green blobs are the cell bodies of these cells, and you can see that they sit at the interface between the pain circuitry and the innocuous touch circuitry. And essentially what we found recently is that when you apply particular types of noxious stimulation to the hindpaw of anaesthetized animals, these calretinin expressing cells are preferentially activated in response to largely pinch stimuli, but they also respond to capsaicin application as well. More recently, we've been able to selectively activate these cells using optogenetic approaches, where calretinin cells express the light-sensitive ion channel rhodopsin. And what we've been able to do is to photostimulate these animals in vivo, and they actually show behavioral responses. So we have a light probe that's situated over the lumbar spinal cord on one side of the animal. When we photostimulate it, the animal shows a nociceptive response. So they'll lift the hind limb, they'll start licking the paw and show exaggerated grooming responses. What we've also been able to show in vitro is that when we target either calretinin neurons themselves or cells within that plexus that don't express this green fluorescent protein, or cells in the pain projecting regions, the photostimulation outlined here by this grey box persists for a period of time. But as soon as that light stops, we see an increase and a prolongation of activation of these neurons in the various regions. So what we've proposed is that these calretinin expressing excitatory interneurons act as an amplifier for noxious information that's projected into the spinal cord. When we're looking at the effect of administration of morphine, we see that these nociceptive responses are drastically reduced in a dose-dependent manner, which strongly suggests that it's a pain type cursor that these animals are responding to. And when we do conditioned place aversion testing, where we have the animals in a 2-chamber setup, there's no light activation here, so they spend equal amounts of time in both chambers. When we designate one chamber as being the light on chamber, we photostimulate these animals, and very quickly they move to the light off chamber. And when we take them out of these chambers and reintroduce them an hour later, they still remember that this isn't a nice place to be. And then 24 hours when we again reintroduce them into the chamber without photostimulation, they still remember that that's not a happy place and spend more of their time in that region. So by using these types of paradigms, we're able to appreciate the importance of these excitatory cells. But actually, which circuits do they engage with? So what we've done is terminally anesthetized animals and photostimulated them, and then look for markers of cell activation, such as CFOS. And then what we've done is to take a variety of different neurochemically defined populations and to work out whether we see the expression of CFOS within these cells. And that's allowed us to try and paint a picture of how these calretinin cells hook up together. And essentially what we see is that they will talk to each other. They will also activate a population of excitatory interneurons that express somatostatin. And both the somatostatin and the calretinin cells also talk directly to the lamina 1 pain-projecting neurons. So I've outlined and showcased some of the work from my laboratory as an exemplar of what we can do to try and understand circuitry in the spinal cord in regions that receive information from somatic afferent fibers. But what about visceral inputs? Well, visceral inputs are much more difficult, I think, to track, and I think it's fair to say, with the greatest respect, that our understanding of central circuits that respond to different types of visceral inputs is very much still in its infancy. So when we compare the morphologies of individual fibers that have been impaled from skin regions to the central arbors of those derived from visceral afferents, you can already see that there's a huge difference in the complexity of the central termination patterns of visceral afferents. And that increases the challenge that we face. Also, when we actually apply noxious visceral stimuli and look at the expression of activation markers along the length of the spinal cord, we see that there are different spinal segments that are recruited, but also a difference in the distribution of central activation as well. And how do we tackle that? Because that's the main aim for what we want to know now. Well, thankfully, over the last couple of years, work by Jim Hockley and Ewan Smith have been able to do transcriptomic studies on visceral DRG directly, and have been able to identify 7 distinct types of visceral afferent fibers. And that's then led to be able to untangle these circuits a little bit more. And hopefully, with a basic understanding of the template that we see for somatic processing, that we can use that as a guide that will help us understand how visceral information is processed at the very important node within the spinal cord. And will hopefully give us a better opportunity to try and understand how this information is relayed up to the brain for ultimate pain perception. To finish off, we have myself on the way to a Kenny Rogers lookalike competition over here. But also Olivia and Kieran, who are my PhD student and postdoc, and Brett Graham, who's one of my main collaborators as well. The Spinal Cord Group in Glasgow are a great team to work with as well as collaborators with David Ginty, Masahiko Watanabe, Uli Zeilhofer and Hendrik Wildner. And this wouldn't be possible without funding from the BBSRC, the NHMRC and the NC3Rs. And just to finish off, I would like to invite you up to Glasgow for the FENS 2020 meeting, but as I've mentioned, the weather is a little bit changeable. Even though it's July, remember to pack your shorts, but also your waterproof sunwellies. Thank you very much.
Thank you, David. Questions? Comments? Yes.
You mentioned about the differences in the primary afferent innervation. What's known about the differences in the subtypes of interneurons sort of along the length of the spinal cord? Is there very much known there?
I'm not too aware of that. There are neurochemically defined populations that you can see expressed along the spinal cord, but that's not necessarily a guarantee that that defined population in L2, in L3, L4 and 5 are doing the same sort of thing in these different segments. So the basic neurochemical markers that we have is a way of identifying these populations, but whether they serve the same functions in different spinal cord segments or different brainstem regions is still to be determined.
Yes, at the back there.
So just carrying on from that, I mean, you have your theories on this gating and alteration of in neuropathic pain of large myelinated fibres, but of course the gut hasn't got those. So in that case, you imagine the entire circuitry is going to be radically different from what happens in neuropathic pain.
Absolutely, yes, yeah. We still need a basic understanding of the anatomy and the connectivity of these visceral afferents. Jim's work has highlighted that there are at least 7 transcriptomically distinct populations, and each are likely to serve slightly different functions, and to get an understanding of their basic circuitry is probably the first challenge that we face.
Visceral pain in the clinic is usually spontaneous and unpredictable, whereas in experimental settings you have a very clear trigger. Do you think you could model that? Unpredictability and spontaneity?
Yes, I mean, again, the spontaneity will depend, or the spontaneous activity will possibly depend on the types of afferents, so not necessarily all of them will be recruited spontaneously. In peripheral nerve injury, for example, you see greater ectopic firing in the larger myelinated afferent fibres than you do from the unmyelinated C fibres, and it remains to be determined whether there are similar patterns for the different visceral afferents as well.
One last question.
So, lamina 10 is kind of classically seen as the sort of equivalent of lamina 1 for visceral afferent, and you can see in the picture you showed.
Absolutely, yes.
I mean, you
Yep.
do get termination there. Is any of your work, have you actually ever recorded from lamina 10?
Not from lamina 10, no. No.
I guess, from your
Yes, yeah. So recording from lamina 10 is great, but it'd be even better if we have a specific neuronal population that we can target repetitively. If you're going in blind, for example, in a wild-type mouse, you have no consistency of the target that you're recording from. So at least if we have these molecular cues that allow us to look at these populations time and again, we get a better understanding of their role.
But it is at least feasible.
I would think, yes, yes. Yeah, I would think so. Yes, yeah, absolutely.
Thank you. Sorry,
Thank you
we
very
need to
much.
move on.
Not
but
at all.
keep
Thank
talking.
you.